So now we're going to move on to the second part of Chapter 5, and this focuses on two major concepts. So the first is the inheritance of mitochondrial genes, and the second is a phenomenon referred to as gene linkage. So the learning objectives for this chapter include describing the mode of inheritance of a trait that is influenced by a gene that is located on mitochondria. And we're going to explain how mitochondrial DNA differs from what we've been covering for the majority of the course so far, which is nuclear DNA. We're going to explain how linked traits, so traits that are influenced by genes that are linked with one another, are inherited differently from those we discussed in Chapter 4. So traits that are influenced by Mendelian inheritance, so our complete dominance or recessive patterns. We're going to discuss the relationship between meiosis and linkage, and then explain how linkage provides the basis for scientists to create genetic maps and things that are called genome-wide association studies. So Chapter 5 completely focuses on these variety of phenomena or processes that end up producing these inheritance patterns that differ from those that Mendel observed in his experiments. So the things that we have been covering so far are far more complex than just the simple patterns of complete dominance and recessive allele relationships that we covered in Chapter 4. So what ends up happening, as we've discussed, is that the phenotypic ratios that we would expect, given simple Mendelian patterns of inheritance, are completely altered. And so just as a reminder, instead of seeing our 3:1 phenotypic ratios, where the individuals with homozygous dominant genotypes and those that are heterozygous share the same phenotypes outwardly, we've moved on to these patterns where almost always we see instead a 1:2:1 phenotypic ratio in the offspring. So our heterozygotes end up having a phenotype that looks completely different than either of the homozygous genotypes. But we've also learned that Mendel's laws still apply in these cases. Even though it may look like they don't, we still see the key things of the pairs of alleles separating or segregating during gamete formation. Only one allele is still passed on, and they're doing so independently. Well, in the remaining sections of this chapter, we're going to explore two additional ways that genes are transmitted that both have unusual inheritance patterns. So they're different than the ones that we have seen with simple Mendelian inheritance. And those are the inheritance of mitochondrial genes and linkage. So first we're going to cover patterns of mitochondrial inheritance. And mitochondria, as you remember from Chapter 2, are organelles, and they are located within the cytoplasm of cells. And their job is to serve as the powerhouse of the cell. So they provide energy to the cell via cellular respiration. And what cellular respiration involves essentially is harnessing all of the energy that's associated with bonds that hold the large macromolecules that we eat together And it's changing the energy associated with those bonds into ATP. So on the cellular level, that's what respiration is. And it does that by using a molecule of oxygen as an electron acceptor. So the number of mitochondria in a cell actually depends on the cell type, and it can vary from just several hundred to upwards to 1,000, depending on the cells and how much energy those cells need to function. So mitochondria are also unique because as an organelle, they have a double membrane. So they have an outer membrane that serves to separate the actual components of the organelle from the cytoplasm. So that's the thing that provides the kind of encasement for all of the other internal structures of the mitochondria. And then they also have an inner membrane that we can see here. And rather than just being straight, it's folded and all kind of convoluted to create these things called cristae, which are the folds. And that is where cellular respiration actually takes place. So within the very center of the mitochondria, because we've got these two membranes, we have the thing called a matrix. So the matrix and the cristae work together to allow for cellular respiration to take place. Mitochondria are also very unique as an organelle because they have their very own DNA. And that DNA is located within that very most inner compartment, the very center of the organelle, within that matrix. And we can see it here represented, the tiny double helices that are in purple within the matrix. They also have their very own ribosomes, which are also located within the innermost layer of the matrix. So here we see them represented as the little green circles throughout the matrix. So what that means is they have their own genetic information and genes carried on those molecules, and they have the capability to go through gene expression of that information. They have the ability to go through protein synthesis, transcription, and translation because they also have ribosomes within the matrix. So just a little side note, given that we know mitochondria have their own DNA, have the capability to synthesize proteins, they are thought to have evolved from free-living bacteria that were actually engulfed within a eukaryotic cell. So they have all of the machinery that they would need to actually function, and they're encased within a membrane. So we typically denote mitochondrial DNA as mtDNA in shorthand. And the interesting thing about this DNA is that each single mitochondrion, actually contains multiple copies of a circular little tiny mini chromosome. So if we look in this image within the matrix, we can see that we have many copies of this chromosome. And the chromosome itself is comprised of DNA built from the exact same nucleotides that we saw in nuclear DNA. So our adenosine, thymine, cytosine, and guanine. And it follows the exact same complementary base pairing rules that we saw with nuclear DNA. And even though this mini chromosome is actually a circle or organized in a circular way, within that circle, as you can see in this image, we still have that double helix structure that we've come to expect from DNA. So it's still a double helix, but then it's folded over, connected to make a circle. And you can see here that because it's a mini chromosome and it's just this one circular component, we have 37 total genes within the mitochondrial chromosome. And just a little review here from chapter 9. Remember that our nucleotides are the most basic building blocks of DNA, and these are the same between mitochondrial DNA and nuclear DNA. So we have our deoxyribose sugar, a phosphate group, and a nitrogenous base, our A, T's, C's, or G's. So even though mitochondrial DNA and nuclear DNA are comprised of the exact same building blocks, or monomers, mitochondrial DNA differs from nuclear DNA in several ways. So we've already discussed that the mitochondrial genome is contained on just one single chromosome. So because we're way reduced in the number of chromosomes that we're dealing with, mitochondrial DNA carries just 37 total genes. And we can see this image up on top, the genome is denoted here. So through experimentation, scientists have learned what the different regions or genes actually do. What's their job? What's their role when they're expressed? And they're all labeled accordingly on this little map in the upper right-hand corner. So 22 of these genes provide instructions for making tRNA, or transfer RNA. 2 genes provide instructions for making rRNA. And 13 genes provide instructions for making proteins that are involved in cellular respiration. So what that tells us is that the mitochondrial genes actually encode proteins that participate in 2 main things: transcription and translation, of the mitochondrial genomes, and energy production, or cellular respiration. So these are the 2 main functions of the genes located on the mitochondrial genome. So in addition to the fact that mitochondrial DNA carries far fewer genes than we see in nuclear DNA, all these 37 genes are located within the same closed circular chromosome. So instead of having 23 pairs like we saw in our nuclear DNA, we just have one chromosome and it's circular in nature. All of the genes actually lack non-coding sequences. So when we look at this circle, we don't have all of the extra regions of nuclear DNA that we saw, these introns or excess information of nucleotides that are located between genes. We simply don't have those non-coding sequences. And some of the protein coding genes actually overlap. So we're dealing with just small bit of space. So to conserve that space in a way, we've done away with the excess of non-coding information. And in some cases, the genes actually overlap. Some additional ways that mitochondrial DNA differs from nuclear DNA includes the idea that no crossing over occurs. So we're going to see that the information, the DNA contained within this mitochondrial chromosome is in fact passed on to the next generation of cells and is passed on to the next generation of offspring during fertilization via the egg. But the difference here is that we never see crossing over. And remember that from chapter 3, we learned that crossing over exchange involves the exchange of DNA, or is it the swapping of genes between homologous chromosomes. So we can't have that happen in this case because we're not dealing with chromosome pairs. We're actually dealing with just a single chromosome, even though there are multiple copies of the single chromosome. And remember that crossing over occurred during meiosis. And meiosis is unique because that is the process that ends up producing our gametes. So interestingly, the mutation rate is much higher in mitochondrial DNA compared to nuclear DNA. And that is explained by two different aspects. So the first is that, remember, the DNA itself, the chromosomes, are located within the matrix of the mitochondria. And that's where cellular respiration is occurring. And for that to happen, we need lots of oxygen. So that DNA is constantly exposed to these free radicals because that's where all of them are funneled to. And the exposure to free radicals actually can damage or alter the bonding nature between bases in DNA and can end up inducing different mutations. They're just changes in the underlying genetic sequence. On top of that, so we've got lots of mutations that are happening, there are fewer ways available to repair the mutations in the DNA. So we learned that there are lots of repair mechanisms built into our nuclear DNA. DNA polymerase itself actually can check for errors and repair them. But we don't have those mechanisms within mitochondrial DNA. Another big way that mitochondrial DNA is different is that it is only maternally transmitted. So mitochondrial DNA is passed only from mother to her offspring. This is because when we look at this image on the right, remember that sperm, all they are contributing during fertilization is their nuclear DNA. That's the only thing that they are passing on to this fertilized cell or our zygote. They empty all of their nuclear DNA, the genetic contents, are 23 chromosomes, that haploid information into the oocyte. So the oocyte is the thing or the cell that is going to maintain all of its organelles and its nuclear DNA. It receives the nuclear DNA from the father, but that cell goes on to, once fertilized, go through a rapid series of cellular division to give rise to the developing embryo. So sperm themselves also do have mitochondria and mitochondrial genes, but those mitochondria, as you can see in this image, are located here. So outside of the nucleus of the sperm cell, because their function is solely to power the flagella, to power the movement of the sperm. So that mitochondria does not enter the egg. So in this picture, you can see we've got our oocyte with lots and lots of copies of mitochondria. That's because we need all these processes to occur. We need lots of energy powering our egg and the divisions that are about to occur. So that mitochondrial genome is going to be passed on only from mother to offspring. So in very rare instances, because sperm do have mitochondria, some of those mitochondria are actually passed on to the oocyte, but they're actually destroyed during development. So a father is never going to pass on his mitochondrial information to the next generation of offspring, only the mother. So because mitochondrial DNA is only maternally transmitted, it creates some very unusual inheritance patterns where the phenotypic ratios that we would expect to see when we're looking at the phenotypes of a mother and the phenotypes of a father, and we do our Punnett square crosses and we would expect to see certain phenotypes within the offspring, we actually don't see those. They deviate from what is expected because we're only seeing genes passed down the maternal lines. So this shows you a pedigree that demonstrates mitochondrial inheritance and how this looks very different. So the first thing to note, because mitochondria are maternally inherited, mothers pass on the mitochondrial disorders to all of their offspring. So we are going to learn in a few slides that sometimes this isn't the case due to a thing called heteroplasmy, and that gets more complicated. But in general, if a mother has a mitochondrial disorder, she's going to pass it on to all of her offspring. So here we can see this mother, number 2. She's affected by some type of mitochondrial disease. We know this because her symbol is a solid color. And you could see that she has passed this disorder onto every single one of her offspring in the next generation. Each of these individuals, all 5 of them, are also affected. You will also notice that male and female offspring are equally likely to be affected. So the mother passes the condition on. She's the only one that passes it on, but males and females are equally likely to be affected if they inherit the mutations. The final clue that tells us that this pedigree is explaining the patterns of mitochondrial inheritance are the fact that affected males are never passing on the disorder. Right? So we look in each case here, each of her sons, 1, 5, and 9, are all affected by the disorder, but none of their children are affected in any of these cases. Because each of these sons' wives do not have the disorder. So it's never going to be passed on from an affected father to his offspring. So I've already mentioned that mitochondrial genes encode proteins that participate in 2 main things. One is protein synthesis, because we've got effects of on tRNA and rRNA, then also energy production. Those are the 2 things that are happening. So there are several diseases that can result from the mutations that actually occur in mitochondrial DNA. So some of those diseases relate to the fact that the mitochondria, their job is energy production. And these diseases are classified as mitochondrial myopathies. And the symptoms associated with these myopathies relate to the fact that there's typically fewer energy available in the cells. And they also occur in cells that normally have lots of copies of mitochondria. So I mentioned that different cells have different actual concentrations of mitochondria. So we often see these disorders in skeletal muscle because, as you can guess, our muscles require lots of energy to function. And because of that, they have many copies of mitochondria. And so when we see these diseases appear, they're associated with just an overall fatigue, or individuals with the disorders become fatigued faster than other individuals. They simply don't have any energy. The muscle structure and appearance itself changes, so muscles become weak and flaccid, unable to perform regular activities that a healthy skeletal muscle would. Another example of a mitochondrial disorder is Leber optical atrophy. So it actually can cause impaired vision. So the muscles that help the eye function, if there's a disorder associated with a mutation in the mitochondria that it can't process the energy the way that it could, those muscles become weakened and lead to impaired vision. We also see mutations in the genes that encode the tRNAs or the rRNAs. And that can cause problems because it directly affects the organelle's ability to manufacture just any of the proteins that are encoded within the genome. So interestingly, there's a transfer technique that if there's a woman who has a devastating mitochondrial disease, and doctors and scientists are aware of that, they can actually perform this transfer technique called an ooplasmic transfer so that the mother will avoid transmitting the mitochondrial disorder onto her offspring. I don't know much about this technique, but it's interesting that there are options to deal with these types of disorders. So this slide shows a better representation of how heteroplasmy influences inheritance. So just a reminder that heteroplasmy strictly refers to the fact that some mitochondrial chromosomes carry a mutation. And in this case, those mitochondria are colored in red, while others have chromosome copies that are completely wild type. And those are depicted in green, so no mutations at all. So in this scenario, we're going to start with a mother who has either mild or no symptoms associated with a mitochondrial disorder. So how can we start with a mother that isn't showing symptoms and end up with children who actually have severe cases of the disorder? Or the other way around, we can have a mother who is showing mild symptoms or no symptoms associated with the disorder and also have offspring that show the same phenotype that she does. So looking on the left, we're going to start with a scenario where the mother cells have about 20% of these mutant mitochondria. So overall, 20% of the total mitochondria in all of her cells are carrying a mutation. So remember that each individual mitochondria has multiple copies of the mitochondrial genome. So they have multiple chromosomes within them. And typically we start with maybe a few of these chromosomes have a mutation and some don't. So what ends up happening for whatever reason, and I still don't completely understand this and can't find literature to back it up, But over time, the chromosomes within each of the mitochondria end up being dominated by either all of the wild type or all of the mutants for any particular gene. So the mitochondria themselves, however, are going to be likely different with regards to the allele that they carried for the particular gene. So that's why we're using this red and green coloration. So we start with this mother who's 20% mutant mitochondria within her cells. So remember that we all start as a zygote. The egg and sperm come together. We begin with this cell in the beginning, which undergoes lots and lots of rounds of mitosis to ultimately give rise to the specialized cells that go on and become all of the cells in the different parts of our tissues and our bodies. So in the very beginning, this is sort of representing how she starts off with 20% mutant mitochondria, but the cells that are going to go on and become eggs eventually, remember these are oogonia that already are starting to form during development, they end up just randomly receiving a different proportion of these mutant mitochondria. So you can see how, depending on how we select the number of mitochondria that are going to end up moving into these oogonia, we can have cases where some cells are dominated by these mutant mitochondria and randomly other cases where it's dominated by the wild type. So these oogonia have to go through the 2 rounds of meiotic divisions, and as they do so, the mitochondria within them increases. So before any of these cell divisions, during the cell cycle and interphase, the organelles are always copying themselves as well. And that's important because the cells that will eventually become these mature egg cells, or our oocytes, need to have lots of copies of mitochondria to produce energy and to properly function. So what we see is that the proportions of these mutant mitochondria stay about the same, even though we have lots more of them. So randomly, as these oocytes mature, we've got these egg cells that are ready to be fertilized. The number of the mitochondria can actually drop to about 100 or fewer. And so that's what we're representing in this case. And if the mother was originally had a replacement for the mutation, as we see here with her 20% mutants, by chance, she can end up producing a variety of oocytes. So you can see that she may have some oocytes that are dominated by these mutant mitochondria. In this case, we've got 80% of the mitochondria are carrying a mutation versus, say, 50% versus 20%, just as the rest of her cells are. So this makes it possible for this mother who doesn't have symptoms or maybe mild symptoms associated with the disease to go on, give rise to these oocytes that actually are dominated by mutation-carrying mitochondria. And if those are randomly, or that is the randomly fertilized egg cell, in this case on the top, she ends up giving birth to a child who has severe symptoms associated with the disorder. And our other example, way at the bottom, is there's also an equal likelihood, because this is all random, that she may have a child that is similar to her. Cells where you just have 20% of these mutations and that individual doesn't show any symptoms associated with the disorder. It also depends on where the affected cell types, or which cells go on to be the ones that end up containing these mutated mitochondria. So the previous slide showed how these conditions are passed on, mitochondrial disorders are passed on from mother to offspring. And this slide shows you how heteroplasmy can then go on to have consequences for phenotypes. In other words, it directly affects expressivity. And so we've already covered what variable expressivity is. And in the first part of this lecture, remember that it relates to the severity or extent of a phenotype. So in this case, depending on the number of mutant copies of those mitochondria within cells, and this also depends on what type of cells that we are actually dealing with, the severity of the disorder can be extremely variable even among offspring. So in this scenario, and this is an image taken from your textbook, we start with a mother who has this, say, 20% proportion of mutant mitochondria. And after we go through meiosis, giving rise to oocytes, as we just saw in the previous slide, we're now looking at what happens during development. So if just by chance we've got an oocyte that contains 20% of these mutant mitochondria, the one that ends up getting fertilized, goes on to give rise to muscle cells, we have the scenario on the left where all of these cells still contain a few copies of the mutant mitochondria, but they're few enough that the issues associated with their dysfunction isn't going to be realized as symptoms because we have enough of the wild type copies that are able to function properly. But there is a threshold, and once we're past this threshold where, say, the oocyte that was the one just randomly ended up getting fertilized, and we've got a greater proportion of these mutant mitochondria, we'll say 40%, that's the oocyte that got fertilized, and just by chance, the way that the mitochondria within that very first zygote that goes on to give rise to these specialized cells, the way those mitochondria are distributed ends up being more and more of them in, say, the muscle cells of the offspring. That's where we're going to see the greatest effect. So you can see here across the bottom, we are showing the severity of the disorder. So that's our phenotype, the symptoms associated with the disorder. And moving from left to right, the severity increases. So the symptoms would be the same associated with that mutation. Let's just say that there's not enough ATP or energy produced, and it causes fatigue. Well, an individual that's just above the threshold may become fatigued, but it may take a greater amount of time, say more exercise, more activity before they feel those symptoms. Maybe they just feel a little bit tired. Versus on this far end of the schematic, we would have an individual who could become severely fatigued after just, say, walking a flight of stairs. So you can see how the proportion of mutant mitochondria relate directly to expressivity or the extent to which the symptoms associated with the disorder are realized. So mitochondrial DNA can also be used to reveal events that have happened in the past or reveal connections between individuals, so hereditary connections between individuals. And in addition, beyond just humans, can be used to look at evolutionary relationships between species. And remember that these genetic instructions are only passed from mother to offspring. And so essentially we have a wonderful record of information connecting the maternal line across generations of time. So with regards to forensic science, mitochondrial DNA can be extremely useful, and that's mostly due to the fact that it is more likely to remain at a crime scene or just environmentally than nuclear DNA. And that's because we've already discussed how cells have lots and lots of mitochondria. So there are lots of copies of the genetic information available to remain over time. Whereas nuclear DNA, it's not as abundant and can be lost faster than the mitochondrial DNA. So scientists will use this to link suspects to crimes, to identify victims. When we have cases where we have a body, we're unsure about the identity of that information, the individual, and they can also use it to support or challenge historical records. So the example provided in your textbook discusses how Marie Antoinette had a son, Louis XVI, who supposedly died in prison. And once her son was buried, there were many people who thought that the child buried was actually an imposter. But when scientists more recently found the heart belonging to this son, because it remained in possession of the royal family, they were able to sequence the mitochondrial genome and determine that it was indeed the prince. So now we're moving on to the second topic of the lecture, another phenomena that influences expected phenotypic ratios, and that is linkage. So remember that the genes underlying the phenotypes that Mendel was studying in pea plants, they were located on different chromosomes. So they were always inherited independently of one another, the phenotypes. And he demonstrated that by his dihybrid crosses, looking at two separate traits and how those traits were inherited across generations of time. So genes that are located very close to one another on the same chromosome are actually what we called linked. And when genes are linked or located within close vicinity to one another, they absolutely cannot assort independently during gamete formation. So remember, Mendel's second law was that alleles segregate independently. And because those alleles segregated independently, the phenotypes that he was studying were not associated with one another. Well, in this case, when you do have genes that are located within close positions along the same chromosome, they are going to be inherited together as a packet. So essentially they're packaged together into the same gamete. And there's no way for crossing over to separate them because they're located so closely to one another. And what that means is that they're always dependent upon one another. And we see certain combinations of phenotypes always occurring in the same way across generations of time. And what that does is alter our expected phenotypic ratios. So here on the bottom of the slide, you can see what I'm referring to when I say linked. So we've got two genes, and linkage relates to the scenario on the left where we have two genes. Their loci or positions on the chromosomes are within very close vicinity to one another, and what that means is they're going to always end up being packaged together within the gamete. And so they're always going to be passed along together in the same combinations from parent to offspring. The second image is where we've got genes that are not linked, and that's because they are on completely separate chromosomes. So that represents the case where that Mendel was dealing with, because all of these genes were on independent chromosomes. They can never be packaged or inherited together because they're independent of one another. Now, we can have cases where we have genes on the same chromosome, but they still are inherited independently of one another because they occur far enough away from one another on the same chromosome. So they're not always going to be packaged as this combination because crossing over can separate them into different gametes. And we'll explore how this works in the next few slides. So this image is taken from your textbook, and it demonstrates how genes that are close to one another are inherited together as a package. So it demonstrates why these genes are linked. So in this scenario, we're dealing with two different genes located on the same chromosome. So we have our P gene and our L gene. And remember that during metaphase 1 of meiosis 1, our homologous chromosomes pair up in the center, because the goal of the first round of meiosis is to separate these homologs. So we also can tell that our chromosomes are represented by these two identical sister chromatid copies because we've already gone through replication during interphase. So the individual that we're starting with here is heterozygous for both of these genes. So you can see that for the P gene, they have one dominant and one recessive P, so they are PP. And for our L gene, they have a dominant and recessive L. So the genotype, considering both of these genes, is PP and LL. So we're going to separate our homologs during meiosis 1. That's what you can see here. We're passing on only one homolog to the gametes, the other homolog in the other direction to our gametes. So here I'm separating the maternal and the paternal contributions, these chromosomes. And what you can see is that these genotypes remain the same. They haven't changed. So in one case, we're passing on our dominant P and our dominant L, half of that genotype. In the other case, we're passing on our recessive P and our recessive L. So the two dominant alleles are always going to be passed on together as a package. And the two recessive alleles are always going to be passed on as a package. So what that means is you're never seeing a gamete that's going to have a dominant allele for one gene and a recessive allele for the other gene. Dominants are always together, recessive are always together, and that's because the locations of those genes are so close, whatever they are on the homolog is the way that they're going to be passed on to the gamete. So you can see down here, we go through a second round of meiosis, and we're separating those sister chromatids from one another. So we end up producing gametes that either have dominant P, dominant L, or the recessive alleles for these genes, recessive P and recessive L. And that's it. That's the only combinations that we have. They're always packaged together in the same way. So this slide actually demonstrates how linkage can alter the expected phenotypic Mendelian ratios. And scientists Bateson and Punnett actually first observed these unexpected ratios that indicated that linkage of genes actually occurred with some experiments that they were conducting on pea plants, and the phenotypes that they were investigating were flower color and the length of pollen grains. So what we can see in this image, we're going to walk through a dihybrid cross. And remember that dihybrid is referring to heterozygous genotype for two different genes, because we're looking at two different traits. So in both of these scenarios, the genotypes of the parents are actually exactly the same. Right? So we look here and we've got heterozygous genotypes for two different traits, and they're exactly the same in both scenarios. So we've got big P, little p, and big L, little l. So the individuals are heterozygous for both genes. But the key is where these alleles are actually located. So in the expected scenario, our true dihybrid cross here, we're going to cross two parents that are heterozygous for both genes. And you'll notice that for this to actually work and the alleles to be segregated independently into the gametes, we're dealing in this scenario with two different chromosomes. So our P alleles would be on one chromosome, and our L alleles would be on a separate chromosome. So the parent is heterozygous for both of these genes, but the key is that the potential gametes that the parents can make actually have way more potentials than what we see when the genes are linked. And that's because during the first round of meiosis, we're going to separate each of these homologs. I'm going to separate our dominant P, our recessive P, and our dominant L, and our recessive L. And because we separate them and the way that they align during metaphase is completely independent, we then can end up producing 4 different combinations within our gametes because they're independent of one another and be packaged independently in the gametes. So we can create some gametes that have both dominant alleles for the genes, PLs, a dominant and a recessive allele for our L, recessive P, dominant L, and both recessive P and L genes. And we can do that for both female and male gametes because remember, both parents are hybrids for both phenotypes. And when we do this, that gives us our classic expected 9:3:3:1 phenotypic ratio. And you can see the way that everything combines here in our giant Punnett square. But the point is, because these alleles are located far away from one another because they're on different genes, on different chromosomes, they can be inherited independently. So we see this great range of phenotypic variation within our offspring. Now, when scientists were conducting these experiments, they realized that in this case, Bateson and Punnett found that they were not observing some of the phenotypes that they actually expected to. So you'll notice that there are 6 different phenotypic classes here that are completely missing in the scenario on the right. So we're not seeing any of these phenotypes that are in these blues or greens. And that's because those phenotypes are combinations that we're simply not getting. So if we do this dihybrid cross on the other side, but our P and L genes are actually linked, so remember the genotype is exactly the same. But because these genes are located close to one another, and we already walked through this on the other slide, the only way for the alleles to be packaged into the gametes are 2 different options. We're always going to package the 2 dominant alleles together and the 2 recessive alleles together, instead of getting this nice array of combinations that we saw over here. So instead of 4 possibilities, now we only have 2 for both the parents because they have the same genotype. And when that happens, you end up in our Punnett square, we get 4 different genotypic, and 3 different phenotypic possibilities. So we only have 4 different options for our genotypes, where we have 4 different genotype combinations, whereas in our classic dihybrid cross, we actually have 16 different options. And instead of having 4 different phenotypic possibilities, 4 phenotypic possibilities, on this side, when the genes are linked, we actually only have 2. So we end up reducing the different potential genotypic combinations that we'll see in our offspring and the phenotypes that we see in our offspring. Classes that we expected to see are completely missing. So we now have a 3-to-1 phenotypic ratio when really we were expecting a 9-to-3 to 3-to-1. So our potential combinations are completely reduced, and that's because the alleles are always inherited in a dependent fashion of one another, always as a package rather than independently. So remember from chapter 3, when we were covering meiosis, we discussed this phenomenon or process called crossing over. And that happened during prophase 1 of meiosis, and it occurred between our homologous chromosomes as they were paired up during prophase. It was because they were paired up so closely to one another, they were actually swapping genes. So the chromosomes are recombining or exchanging genes during prophase 1, and that's called crossing over. And when crossing over occurs, what that does is it gives rise to genetic diversity. And how that exactly works all comes down to the fact that we're creating new combinations of alleles. So we're not creating new alleles, but we're combining our alleles a different way than what we started with. So a parent may have a certain combination of alleles, but they can pass on a different combination of alleles to their offspring. So the parental chromosomes that we start with have that original configuration. So the way that those allele combinations are situated on a chromosome. But when the homologs actually exchange genes, it creates new combinations, and those are called recombinant chromosomes. It's what ultimately gives rise to one source of diversity associated with meiosis. And this crossing over can actually disrupt linkage. So because we are able to create new allele combinations, those alleles are essentially being inherited independently of one another. They're not always going to be packaged the same way like we saw with linkage. So here we can see we're dealing with our P gene and our L gene again. But in this case, these genes are, even though they're located on the same chromosome, they're actually located pretty far apart from one another on the same chromosome. So when genes are located far enough apart from one another, even if they're on the same chromosome, they can behave independently as if they were on separate chromosomes. So what you can see here is during prophase 1, our homologs are going to swap genetic information. And remember that the homologs are actually overlapping on average up to 3 different regions across the legs of the chromosome. So lots of swapping going on. But in this case, we're just focusing on the 2 genes. So what can happen, as we see here, if we swap and move this P, our recessive P, to where our dominant P was, and that's the only spot that we even switch. We're not even dealing with switching the alleles on the L gene. So just one simple swap of an allele ends up creating 2 recombinant chromosomes. Because now instead of having our 2 dominant P and L alleles together, one of our homologs has a recessive P. And so it's going to go with the dominant L, and the other homolog gets the dominant P. So now you can see, instead of the only possibility of having our dominant alleles together and our recessive alleles together being packaged together, always passed on to the gametes, crossing over now enables us to actually have two different combinations. So we can have gametes that inherit a recessive P and a dominant L, and a dominant P and a recessive L, as well as the ones that we expected. Our two dominants together and our two recessives together, the same way that we observed it in the parents. So half of our gametes are going to have the same allele combinations that we saw in our parent. So these are our parental chromosomes. Just as we saw in the parent, we have our two dominants together and the recessives together. But we now have a recombinant chromosome that's being passed on to the gametes where you can have a recessive and dominant together that we didn't see before because of crossing over. So these genes are not linked because they're not always inherited as the same package. They can be inherited independently from one another. We can see new combinations arise in the gametes than what we saw in the parent. So allele configuration, or the locations on the homologs, is really important in terms of being able to determine if we're producing or dealing with recombinant chromosomes or not. So it's all about where the dominant and recessive alleles are located with respect to one another when dealing with these two genes. So in our first scenario, which is what we've been looking at the past several slides, is called cis configuration. So our dominant alleles are always occurring together on one homolog, and the recessive alleles are on the other homolog on the parent chromosomes. We can detect recombination because the way the alleles then are packaged or mixed up is different than we're ever going to see on the parental chromosome. On the other hand, if we're dealing with transconfiguration of these alleles, on the parent homologs, we have one dominant and one recessive allele, just like we saw with our recombinant chromosomes. So it makes things more complicated. No, this makes no sense. So in order for us to determine if recombination is happening, we really need to know the allele configuration of the parental chromosomes. So the case that we've been dealing with for the past two slides, we have our alleles configured in a cis formation. And what that means is that our parental alleles were always together as dominants or recessives on the same homolog. And then we knew if in the offspring we saw a dominant and a recessive allele on a homolog, that recombination had occurred. So these alleles can also be together in a trans formation. So in that scenario, we've got one dominant and one recessive allele on each parental chromosome. So the genotypes are the same for both these scenarios. We're heterozygous for both genes, but the way that the alleles are positioned on the homologs is different. So they're either cis or trans. And as long as we know how they're packaged in a parent, we're going to be able to tell if recombination is occurring or not, because we're seeing the alleles packaged in a different way in the offspring. But in order to do that, we need to know how they occur together on the parental chromosomes. So in the 1900s, as scientists were performing these crosses and observing that there were scenarios where the phenotypic classes were not as expected, so they learned about this idea of linkage. The idea for using this information that some genes are linked and some genes are not and can give rise to different allele combinations grew into this idea of creating a genetic map. And creating a genetic map was related directly to the location of alleles with regards to how close or far away they are from one another on the same chromosome. So here we can see that the frequency of recombination, or the extent to which the exchange of genes or allele combinations are occurring, is related directly to the distance between the genes, the distance between the locations of them, or the loci on the chromosomes. And all of that was used as information to construct linkage maps. So here you can see that when genes are farther away from one another, as the A and B gene are here in this case, the likelihood that the allele combinations are going to switch, crossing over is going to occur, and recombinant chromosomes are going to be generated in the gametes, far more likely. Genes B and C, however, crossing over is less likely to occur. So we're actually going to see fewer recombinant chromosomes in the offspring. And when that happens, we know that those genes are closer together. So it's using both of these ideas in tandem. The more recombinant chromosomes you see leads to the idea that, hey, those genes are farther apart from one another, versus where you've got gene combinations and the recombinant chromosomes are quite rare, you know that those genes are closer together. So working together with those two ideas, doing crosses and looking at phenotypic classes of offspring, helped scientists create linkage. So the frequency of recombination can be determined by looking at the percentage of meiotic divisions that actually end up breaking apart allele combinations. So instead of end up being linked with one another, we actually break that likelihood that alleles will always be packaged in the same way. So we're taking parental alleles and we're separating them into different gametes. And the likelihood that that will actually happen is directly proportional to the distance between the two genes that are involved. So these two things go hand in hand. The greater the distance between two genes, the greater the chance of crossing over or breaking apart those allele combinations yields the greater percentage of recombinant chromosomes. So it's all about distance. So this slide demonstrates how even if genes are located on the same chromosome, if they are actually far enough apart, recombination is so frequent that it can create allele combinations in the exact same proportion as we would see during independent assortment. In other words, we can have genes that are located on the same chromosome actually being inherited independently, as if they were located on completely separate chromosomes. So in the top scenario, we're looking at two genes, and they're located on the same chromosome. In the bottom, these two genes are on different chromosomes. So in the top scenario, it's calling them linked because they're on the same chromosome, but I don't necessarily like that term because they're far enough apart from one another, they're always going to be inherited independently of one another, just as we see as if they're on a different chromosome. So you can see here, because they're far enough apart, remember, greater the distance, the greater the likelihood that they're going to recombine or swap locations with one another. So what we see is that in our parent, and both of our genotypes are the same, whether we're on the top or the bottom, this slide. So we've got dihybrid individual, so heterozygous for both genes. Half of the gametes are going to end up with allele configurations, just like we saw. And the other half of the gametes are actually going to end up, even though they inherit the same alleles, they're going to be on different chromosomes, on different homologs. So the alleles are packaged differently. So we've got the chromosomes are essentially a combination of the maternal and paternal information rather than being contained on the same chromosome. In the bottom scenario, same genotype, we're still dihybrid with regards to these two genes. But if the genes are now located on different chromosomes, it all comes down to the fact that we're going to separate these chromosomes independently of one another. during the rounds of meiosis. And it all depends on which side of the metaphase plate that our homologs are aligning. So remember that they pair up and we're going to separate them. And in the first example, here's our metaphase plate. So they paired up and then we're going to split them. We're going to still keep our parental dominant alleles and recessive alleles together, just as we see when we start. But if these homologs reverse in the way that they line up, and we end up with a blue and a green on each side, you can see how the allele combination that's passed on to the offspring via the gametes is different than what we saw with the parent. And that recombination configuration is exactly what we see up on top. So the point here is that even though Mendel's second law of independent assortment related to the ideas that genes on separate chromosomes assort or are separated independently of one another, we can see that exact same phenomenon if the genes are on the same chromosome, as long as they are located far enough apart from one another. So the law of segregation, the law of independent assortment, holds true even for genes located on the same chromosome as long as they are far enough apart. This slide actually walks you through the process of creating a linkage map. So this is just a diagram indicating the relative distance between genes. And we can use the information on the percentage of recombination between genes to figure out how far apart or close together they are. We've already talked about how the percentage of recombination is directly proportional to the distance between the genes. And every 1% of recombination is equivalent to 1 map unit, or a centimorgan. And all of these distances are additive. So if we've got 3 different genes that we are dealing with, we can place them organized pairs, and we can determine the percentage of recombination between them. So in this scenario, X and Y is a 10% recombination, right? X and Z is a 4%, and Z and Y is 6%. So we can take all of that information. We know that because X and Y recombine more frequently, they're the ones that are furthest apart. And then we know that Z is somewhere in the middle of the two, and we can actually even determine that it's closer to X than it is Y based on the percentage of recombination between the genes. So this is the only configuration that we can create that enables for all of those distances to work out in the correct way. So the phenomenon of linkage, the idea that when you have genes close to one another, they end up becoming linked, or they're always inherited as a package, creates this thing called linkage disequilibrium. And that simply refers to the fact that we have non-random association between DNA sequences. So this just simply means they are not inherited independently of one another. They're inherited together more often as a package than would be predicted given their frequencies in the population. And the human genome actually consists of many of these linkage disequilibrium blocks, because we've got alleles that are tending to stick together or are inherited as packages at all times. And then those packages or linkage disequilibrium blocks are interspersed with areas of DNA where crossing over is extremely prevalent. So we can use this information of gene locations with respect to one another and linkage to solve genetic problems. And I am not going to ask you to do this on the exam. I just want you to be aware that we need to incorporate linkage information when we're working through inheritance questions. And that's in all genetics problems. So the example that your textbook gives you is that the genes for Nail-Patella syndrome, which we'll represent as N, and the ABO blood type, I, are 10 map units apart on chromosome number 9. So if I were to give you the following scenario, that Greg and Susan each have Nail-Patella syndrome, Greg has type A, Susan has type B blood, what is the probability that their child has normal nail and knees and type O blood? So we're going to use the information in terms of the allele configuration and the distance of these genes to calculate the frequency or proportion likelihood that their offspring will have normal knees and type O blood. So let's say that Greg and Susan go to visit a genetic counselor because they both have this syndrome and they're interested in knowing what their offspring would have in terms of genotypes and phenotypes. So the counselor can create a modified pedigree by asking information about Greg and Susan's parents. So Greg's mother also has Nail-Patella syndrome and type A blood. So because Greg has both of those phenotypes, that means that his father must have normal nails and also type O blood. So what that means is that Greg inherited the dominant Nail-Patella syndrome allele, because this condition is dominant, and the A blood type allele from his mother. And the other alleles on his genotype then come from his father, so the recessive N and the recessive I, which is what we use to denote type O blood because there are no antigens. So that means for his allele configuration, we know that he inherited the two dominant alleles, the N and the I, A allele from his mother, and the other allele combination comes from his father, so the recessive N and recessive I. So the genetic counselor also asks about Susan's family history, and we find out that her mother also had Nail-Patella syndrome and type O blood, but her father has normal nails and type B blood. So here we can see that her alleles, in contrast to Greg, are actually in the transposition. So we've got the one dominant allele and one recessive allele of the other gene being packaged together. So now we need to work out how these alleles would end up being packaged in the gametes, the sperm and the oocytes, and thinking about any recombinants that also might occur. So we're asking specifically about the probability that the offspring is going to have normal nails and type O blood. So the only way we can get this genotype is if a normal nails and recessive I, or type O allele, which is right here, were to combine with another normal patella allele and recessive I oocyte. So what we see is this is the way we want our gametes to unite. We want to know first we'd have to determine the frequency of these gametes. So you can see that Greg is more likely to produce this normal allele combination, if you will. Obviously, O blood type isn't normal, but these two recessive alleles, 45% of the time, we're going to see the alleles packaged in this conformation. And that's because half of his genotype is actually coming from this allele combination on the parental homolog. So 45% of the time he's going to produce this parental sperm. For Susan, however, that combination is much rarer. And that's because the only way we're going to see that combination is if crossing over happens and we have recombination. Give rise to a new allele combination of the normal and recessive I allele. So, because these are independent events, we can multiply them. So we would take the frequency of this sperm, likelihood, I skipped ahead, come back. This frequency here of 45%, multiply it by the frequency of that egg combination, which is only 0.05, and that gives us the final value of 0.025. Convert that into a percentage, which is quite low, so it's only 2.25% chance of giving birth to a child that will not have the syndrome but also have type O blood. Calculating other genotypes or addressing other questions that we may ask is going to be even more complicated because more combinations of sperm and oocytes could account for them. So, for example, if a child with Neal-Patella syndrome and type AB blood could arise from different combinations, several different combinations, and we'd have to figure out the likelihood for all of those events. So this is the simplest question we could ask, the event of not having Neal-Patella and having type O blood. So how do we get from linkage to genome-wide associations? Well, the first human genes that were mapped onto their chromosomes included blood proteins. And that's because they were pretty easy to study. And in 1980, researchers began using DNA sequences that were located near genes of interest as landmarks. And they refer to these as genetic markers. So if you work in a lab or you've read genetic studies, you'll frequently hear this term genetics markers. And they just denote the location of a particular gene of interest that scientists are studying. So these markers don't need to encode proteins. that are associated with a phenotype. They actually might be DNA sequences that essentially differ with regards to a cutting site from an enzyme. And so when that happens and the enzyme is cutting at different locations, you end up with different lengths of little DNA sequences associated with the gene. So they don't necessarily perform a function, but they're just a cutting site. And the term genetic marker has come into use and in popular science because it usually refers to a sequence that's associated with a particular phenotype, especially one that affects an individual's health. And these genetic markers are more recently being used in genome-wide association studies. So scientists can actually use computers to determine how often genes of interest and their markers are inherited together. And that's, again, related to linkage, right? Because linkage is related to how close in vicinity to aspects of the genome are. So the LOD score, which stands for logarithm of the odds, I can't talk anymore, indicates the tightness or extent of linkage between the marker and a gene of interest. And it relates to the likelihood that particular crossover frequency suggests linkage rather than just inheritance by chance. So this is simply what we've been covering this whole lecture, the idea that things are closer together, are more likely inherited together or as a package. So to put this in perspective, an LOD score of 3 or higher tells us that there is linkage between the marker and the gene. And what that means is if there's linkage, the data are 1,000 times more likely to be due to the fact that things are closer together than if by chance alone. Another term that we use quite frequently in scientific studies related to genetics is a term called a haplotype. And before any sequencing efforts were done on the human genome, scientists would use genetic markers that could predict which individuals and families were likely to inherit a particular disease. And there are sets of DNA sequences that are inherited on one chromosome due to linkage disequilibrium. So we've already discussed how things that are closer together are linked, and that causes linkage disequilibrium because alleles are inherited non-randomly. And so these sequences that tend to all occur together as a giant package actually made it possible to track specific chromosome segments and pedigrees. And that's because typically the sequences were inherited as this giant package and scientists could follow them through the pedigrees. And if there were any disruptions of the marker sequence or the actual haplotype, it indicated that crossing over was occurring. So this final slide shows you how haplotypes can be informative when looking at a genetic disorder along a pedigree. So here we see that individuals with this disorder have these particular markers associated with an affected allele. So in this case, the father is just a carrier. So one of the alleles, and the marker here is the series of 4 twos. So it's telling us that there's an affected haplotype. For the mother, she has 2 mutant alleles. So we see we've got 2 markers that are associated with the disorder, and she has 2 mutant copies. So we can see that 3 of the offspring are affected, and you can see that all of their alleles and haplotypes are associated with the disorder. However, we have these 2 offspring, male and a female, who don't have the disorder. And interestingly, when looking at their haplotypes, we can learn that they're not even carriers like their father. They've actually inherited 2 functional copies of the allele, and we can determine this based on looking at their marker. In this case, these markers are not what we would expect to be associated with the haplotype. So we're seeing a disrupted marker, and that's completely due to the fact that we had crossing over events between the affected alleles of the mother. And because that has happened, these individuals aren't affected by the disorder, and they're also not carriers.